We're live now.
Okay. I'd like to welcome everybody from the University of York and all the viewers who are coming via YouTube. Today we have a very distinguished guest, Professor Pamela Soltis, who comes from the Laboratory of Medical Systematics and Evolutionary Genetics and the Florida Museum of Natural History at the University of Florida. So Pamela Soltis has had an outstanding career in studying plant diversity and evolution, looking into topics like polyploidy, hybridization, radiations, and many other topics in plant evolution. She's currently the director of the University of Florida Biodiversity Institute and the founding member. She's won numerous awards throughout her career, and I'll just mention a few right now. She's a member of the National Academy of Sciences and the American Academy of Arts and Sciences. She's won the Darwin Wallace Medal from the Linnaean Society of London. She's won the Botanical Society of America's Merit Award. And actually in 2014, Thomson Reuters named her one of the world's most influential scientific minds. And today we have her presenting her most recent research. So I'll hand over to you, Emma.
Thanks so much, Dan, for the introduction, and thank you very much for the invitation to get to talk with all of you today. I almost said good morning, but I realized it's not morning, it's after lunch for you. So I hope you're all well fed and ready to go for the afternoon. I'll go ahead and share my screen and get started. So today I'd like to share with you some of the research that we've been doing on polyploidy over the last several decades. And I like to think about polyploidy as a process that integrates from cells to ecosystems. And although I won't really get into any cell biology or ecosystem level research, I think it will be clear from the material that I present that polyploidy really provides us, though, with an opportunity to extend from the very smallest levels of organization in biology to the very largest. And before I get started any further, I'd also like to note that all of the work that I'll be describing today has been done in collaboration with Doug Soltis, and he's standing here in a field of polyploid cardamine in the Swiss Alps that we visited a few summers ago. Now, there are about 500,000 species of green plants, and they represent nearly 1 billion years of evolution. And although plants have a lot of really spectacular features, I think one of the most amazing attributes is their tremendous variation in genome size, with genome sizes that range from 50 megabases to 150 gigabases. In other words, a 3,000-fold difference in genome size. And in fact, we can see that variation in genome size even within the flowering plants. We don't even have to span all of the green plants themselves. So how does this variation in genome size arise? Well, there are two main avenues. One of them is polyploidy, and the other is transposable elements. And certain groups of plants, such as conifers, have very large genomes due to expansion, particularly of transposable elements. And this is also a process that takes place across many other lineages as well. But polyploidy is something that actually is found throughout almost all groups of green plants. And it's something that has not only implications for genome size, but also for the genetic composition of and genome content. And in many cases, this polyploidy or whole genome duplication can be quite extensive. So in the fern Ophioglossum reticulatum, we see that the haploid chromosome number is 630. So yes, that's 1,260 chromosomes in this little fern. Now, for a little bit more background on polyploidy in plants, I thought I'd share some of the following fun facts with you. There are about 50% of all flowering plants have been proposed to be of polyploid origin. These estimates are based on traditional means such as chromosome numbers and various other sorts of intuitive approaches. that have been conducted over the past several decades. And these estimates actually range from 30% to 70%, but most fall somewhere around 50%. But we'll return to this topic a little bit later. It's also been estimated that about 95% of all fern species may be polyploid. That's also a number that we might want to revisit a little bit later, but I don't think we'll have time to do that today. Maybe that's a topic for another discussion. Also, most of our major crops are polyploid, and some of the most common ones are listed here. And in addition, 17 of the 18 world's worst weeds are also polyploid. This is based on a compendium that was actually published probably about 20 years or so ago. And the 18th one could also be polyploid, but it hasn't had its chromosome number counted yet. Now, in addition to polyploidy that we can recognize easily on the basis of chromosome number, there have been hundreds of ancient whole genome duplication events or ancient polyploidization events that have been identified through genomic methods. And we'll be returning to this topic in a little bit. In addition, some of these ancient whole genome duplications map to the timing of major geological events, such as the KPG boundary. And this work primarily stems from work by Yves van de Peer's lab. So I think you can see just from this brief introduction to polyploidy that polyploidy has tremendous evolutionary, ecological, and also economic importance. Now, I think it's really important to consider the fact that in 1970, Susumu Ono proposed the role of gene duplications as a major evolutionary source of genetic variation. In fact, he considered that gene duplications were really the key source of genetic variation. And that the reason for this is that if you have duplicated genes, those duplicates can diverge in function. One copy can retain the ancestral function, while the other one can be free to tolerate mutations and perhaps take on new functions. So if we think about this perhaps fundamentally important role for gene duplications in evolution, we can then start to, I think, get a good feeling for why polyploidy may be so important, because we're not just duplicating a few genes here and there, but we're actually duplicating the entire genome. Now, shortly after the groundbreaking work of Ono, Don Levin published a paper in the American Naturalist focusing on polyploidy and novelty with an emphasis on flowering plants. And interestingly, in this paper, Levin did not consider the genetic attributes of polyploids, but he focused on those attributes that arise simply from changes in cell size associated with an increased genome size. And he found that there are these so-called nucleotypic effects at the cellular level, biochemical, physiological, developmental, ecological, and evolutionary levels. And that at all of these levels, polyploidy generated novelty. And in a number of cases, he was able to cite examples, but in many others, he proposed some really foundational hypotheses for going forward. And this work was followed up recently by an absolutely astounding review by Jeff Doyle and Jeremy Coate, published in the International Journal of Plant Sciences in 2019, where they revisited many of the themes that Levin raised, focusing again on the nucleotype and on novelty. And they found that, in fact, in the intervening years between Levin's paper and theirs, that many of these questions about nucleotypic effects remain unanswered. And this, in fact, represents a huge open area for future research. So in addition to these effects that are caused simply by changes in cell size, there are, of course, many effects of whole genome duplication that occur at the gene level. So we can have diversity of gene families. There are opportunities for what's been termed subfunctionalization and neofunctionalization, in which the duplicate copies of the genes either parse out the ancestral functions in subfunctionalization, where perhaps a duplicate pair takes on, where one copy will take on a function in one organ and the other copy would take on the function in another organ. Neofunctionalization, in which a new function may arise. And then there can be new combinations of genes. And the underlying point here is really that in addition to novelty generated by nucleotypic effects, there can also be novelty generated from genetic effects. Now, I think as plant biologists, we all recognize the important role that polyploidy plays in speciation. And we can go back to the classic work of those like Klaus and Keck and Heisey, who worked on polyploidy in the California flora in particular, and demonstrated the importance of this as a speciation mechanism. So polyploidy at the speciation level generates genetic and phenotypic novelty as well as new species, but it's also possible that polyploidy can shape macroevolutionary diversification as well. And as I hinted at before, polyploidy can provide an integration across levels of organization. So from this brief introduction to polyploidy, I think it should be clear that polyploidy can have both microevolutionary and macroevolutionary impacts. And for the rest of my talk, the bulk of my talk today, I'd like to focus on these two aspects. First, on microevolution, and secondary on macroevolution, and the sorts of diversification that may arise as a result of ancient duplications. So I'll begin my this part of the talk by focusing on microevolution in the genus Tragopogon. So Tragopogon is a genus that's native to Europe and Asia. Many of you may be familiar with species of Tragopogon, but a few species of Tragopogon were introduced into North America in the early 1900s. And these are the diploid species, Tragopogon dubius, Tragopogon pratensis, and Tragopogon porifolius. And they were introduced into many parts of North America, but they arrived in Eastern Washington state in the early 1900s. And they hybridized in multiple combinations and formed two polyploid species. So Tragopogon dubius and pratensis formed Tragopogon macellus, a tetraploid, and Tragopogon dubius and porifolius formed Tragopogon myrus, also a tetraploid. And this we know this from the absolutely stunning work of Marion Ownby, who was the herbarium director at Washington State College, now Washington State University, and discovered these tetraploid species and named and described them in 1950. Now, since that time, we've learned a lot about these polyploid species. They occur in disturbed habitats, such as along roadsides or railroad tracks. Here's a population that occurs between a railroad track and a road. So this is prime Tragopogon habitat. The polyploids tend to have originated multiple times, basically in all of the small rural communities that occur in this region. So just to get a better feel for the area, this is Washington State up here in green. And if we blow this up, the city of Spokane is located here, and these polyploids originated in this vicinity where the star is. Now, I referred briefly to multiple origins of the Tragopogon polyploids. This is a common phenomenon in polyploid plants. Nearly all polyploids that have been studied in any detail have been shown to have originated multiple times from genetically diverse diploid parental species. So in Tragopogon myrus, up here at the top, this represents a structure plot based on microsatellite data. And each one of these color blocks represents a different population of the tetraploid Tragopogon myrus, and each column represents a separate individual. And what should be clear here is that each population represents a set of genetically identical individuals at these microsatellite loci. but that each population is genetically different from all others, so that we actually have these multiple origins that represent different different genotypes based on different local populations of the parental species. And we see the same general pattern here in Tragopogon missalis, although there's a little bit more genetic diversity within some of these populations. So basically, these multiple origins provide us with natural evolutionary replicates for these polyploid species. So for the past about 30 years or so, I guess, Doug and I have been studying aspects of additivity and novelty in these Tragopogon polyploids. And we focused on at both the gene level and the chromosome level in both natural populations of the polyploids, and also more recently on synthetic tetraploids that we've generated in the greenhouse. And again, these multiple origins provide us with natural replicates. Now, one more little bit of terminology before I describe some of the work that is relevant to this question of novelty and additivity. And this has to do with the term homolog. So homologs are duplicate chromosomes or genes that are derived from different parental species in an allopolyploid. So if these black chromosomes represent the genomic contribution from one diploid species, and the gray ones, those of a second diploid species, the two chromosomes in the circle are homologs relative to each other, but this chromosome pair and this chromosome pair would be referred to as homologs once they are combined in this allopolyploid genome. And all of the genes that are found on those chromosome pairs would also be referred to as homologs. Now we can see this slightly differently in this chromosome painting figure, where we have the dubious genome, the Tragopogon dubious genome, and on the top in green, and the Tragopogon pratensis genome on the bottom here in orange. So here, these two chromosomes from the D subgenome and these chromosomes from the P subgenome would be homologs relative to each other. Same with the B chromosomes, the C, D, E, and F, and so on. So from that perspective, then, using this bit of background of understanding homologs and the subgenome structure of an allopolyploid, we can start to ask questions about whether or not allotetraploid tragopogons are perfectly additive of their diploid parents. And we address this question in terms of their gene content, gene expression, and chromosomes. So first of all, let's look at gene content. And we found very early on in some comparisons that the tetraploids seem to be losing certain copies, certain homologs from their genomes. And so we scaled this up to a larger analysis where we analyzed 450 homolog specific SNPs, and we looked for their presence and or dosage in the tetraploids. So our expectation would be that there would be 2 alleles contributed from dubious and 2 alleles contributed from either porifolius or pratensis, depending on whether we're looking at Tragopogon mirus as up here in the upper left, or Tragopogon missalis as in the upper right. So this is a project that Doug and I organized along with Pat Schnabel from Iowa State University and Brad Barbizuk, also from the University of Florida. And contributing to this work were Richard Buggs, who was a postdoc in the lab at the time, and then subsequently Ingrid Jordan-Thaden, another postdoc, and Lederson Vicini, who was a sabbatical visitor from Brazil. So what did we find? Well, we found that the majority of genes were retained in duplicate. So 72% of all of the genes that we examined in Tragopogon mirus were present in duplicate. And that's what you can see here in the blue bar. That's for natural populations. For a set of synthetic populations, representing the first selfing generation to the fifth selfing generation of the synthetics, we found 83% were in duplicate. But what that means is if those are the percentages of duplicates, that the remainder showed evidence of the loss of one parental copy or the other. So 28% of the natural polyploids showed loss of one homolog or the other, and 17% of those synthetics, which are only up to 5 generations, showed loss as well. And then there were a slight variation in the number of losses of dubious versus the losses of portfolius here on the right. And we see almost identical results for Trichopogon misellus. Very surprisingly, exactly the same number were present in duplicate, 72%, with 28% showing loss, but a slightly smaller number of the synthetics, 80%, were still present in duplicate, with 20% of the loci showing loss. So this is across all individuals. When we look at these multiple origins, these evolutionary replicates, we see very similar patterns, but there is a little bit of variation in that percentage of loss. So it ranges from 65 to 78% of the loci being retained in duplicate, which means 22 to 35% loss. And there's a bit more loss of the portfolius parental copies than there is loss of the dubious copies. And for misellus, again, pretty similar, a bit of variation among the populations, but within the same range. And the loss of dubious alleles or copies is pretty similar to the loss of the pretensus copies here. Now we can look at this in a slightly different way, where we examine actually the impact on each individual in terms of presence or absence of a gene. So this is for Trichopogon mirus, and I'll orient you to how this figure is set up. So we have individuals across the top, we have loci down the side, and so that means we have a square that for each gene for each individual. And if the square is black, it means that there's an additive pattern, both parental homologs are present. If it's pink, it means that, such as here or here, it means there's a loss of a portfolius allele or perhaps entire locus. And if it's yellow, it means that there's a loss of the dubious allele or locus. So that means when we look at this whole figure, the majority of the figure is black. This indicates that both parental copies are present for the majority of individuals at the majority of genes. But we do see these very interesting places where they're across all individuals, or nearly all individuals, there's loss of one parental type or another. So here we see the loss of the dubious alleles in yellow, loss of the portfolius alleles, or maybe entire loci in pink. And so that's true in this block here in the center. And if we look up at the top here, there are a number of loci that have lost the portfolius copy. And this is consistent across all of the natural populations, which are on the left, and across the individuals of the synthetic generations as well. So, so the patterns of loss are not random, and they are often concentrated at particular loci, and that these losses may occur very quickly just within the first few synthetic generations. And we see very similar patterns for Trichopogon misellus as well, with certain loci that have lost one copy, in this case in yellow, again, the dubious copy, across almost all individuals, some individuals actually having lost the pretensus copy instead, but where there's only one copy, even in these synthetics. And then other places where we see similar sorts of patterns. So rather than the polyploids being perfectly additive of their parental contributions, we see that there are many, many instances where one parental copy or the other has been lost. So in terms of whether or not the trigopogons are additive of their diploid parents with regard to gene presence or absence, the answer is no. And essentially every single individual is genetically unique in terms of its combination of alleles across just this set of about 400 loci. So we see novelty arising among individuals. Now, this set of plots is very similar to what you just saw, but this shows data for gene expression instead of gene presence or absence. So this is based on cDNA from the same individuals that were examined in the previous study. And here we see that there's silencing of Trigopogon porifolius in pink for the Mirus example here, for example there, and silencing of dubious in yellow. And there's more silencing of porifolius than there is dubious. And here for Trigopogon misellus, again, similar patterns, certain loci we see silencing extensively. Here for Trigopogon dubious, other loci we see more silencing for pratensis, but overall there's the amount of silencing for dubious is greater than that for pratensis. Now we've also investigated gene expression shifts in more detail using a number of RNA-seq approaches. And the first experiment that I'll describe to you was done by a former postdoc, Mi-Jung Yu, and a former graduate student of Brad Barbazuk's, Lucas Boatwright. And in this example, we included 3 biological replicates of each of the 5 species. And then Lucas developed this pipeline for assembly and analysis. And this was published in Genetics a few years ago. And the basic elements of this experiment are shown here. So we found what we called common orthologous regions or cores based on alignment of the parental reads. So this would show Trigopogon porifolius and Trigopogon dubious. We took the central 90% of these paired sequences and then used these for analysis of gene expression in the tetraploids to see if there was homolog specific expression in these tetraploids. So this shows the results for Trigopogon misellus. In all, we identified about 15,000 of these common orthologous regions in Trigopogon misellus. We did some analyses to test for bias in our read analysis, and we removed about half of the core regions for bias, and that left us with about 7,200 cores for analysis. And of these, about half, or 3,488, showed no homolog specific expression. That is, that both parental types were expressed equally. And then of the remaining half, about half of those showed a bias toward Trigopogon pratensis, and about half showed a bias toward dubious. So basically, half of the loci analyzed showed no homolog specific expression. The other half showed some homolog specific expression, with half bias toward dubious and half bias toward pratensis. And we found almost identical results for Trigopogon mirus. Here we analyzed about 8,000 cores. About half of those had no homolog specific expression. Of the remaining half, slightly more showed a bias toward porifolius than dubious, but those were pretty close to each other. So we do find some evidence for homolog specific expression. That is, the expression patterns are not equivalent between the parental copies in either of these 2 tetraploids. So there's been some shifts in gene expression that have originated even within this very recent time frame of these polyploid these polyploids ages. Now, of course, I think we'd be most interested in examining any sorts of genes for which there is a known phenotypic difference. And so we were very interested in whether or not we could detect gene expression differences between two morphological types of Trigopogon misellus. So I mentioned before that these tetraploids are of multiple origin, but one of the things that's particularly interesting about Trigopogon misellus is that there are also reciprocal origins. So in the case where Trigopogon dubius is the maternal parent and pratensis is the paternal parent, we get what has been referred to as the long-liguled Trigopogon misellus. And when the reciprocal is the case, we get what's called the short-liguled misellus. And these were actually different forms that Ownby identified back in his 1950 paper as well. So we're interested in whether or not we could find any specific gene expression patterns that could be related to this difference in inflorescence morphology between these two reciprocal formations. And this is work that was done by Shengchen Shan when he was a graduate student with us, and this was published last year. So this just illustrates a little bit more the experimental design here, where we looked at expression patterns across the diploids and these two tetraploid derivatives. So we were interested here in whether or not there might be some expression bias in the short and long liguled misellus. And here, this is what no bias would look like if we're talking about an RNA-seq analysis, where there would be equivalent reads mapping to the dubious and the pratensis genomes. And then this would be biased toward dubious, and this would be biased toward pratensis. So what do we see when we actually look at some data? Well, we see that about 80% of the loci examined showed no bias, and the remaining 20% or so are about split between a bias toward dubious, about 8.5 to 9%, depending on whether long or short liguled, and about 10 to 11% bias toward pratensis. So quite similar percentages here between the two parental types. We also were interested in whether or not these diploid or the tetraploids showed differential expression relative to their parents. So one possibility is that the tetraploid M here in the middle shows the same expression patterns as the diploids. Oops, sorry about that. Dubious and pratensis. Here we have additivity, and in this case, in these bottom cases, a number of departures from additivity. So we won't focus much on any of these non-additive expressions, but I just want to call your attention to the fact that there's either no change or additivity in about 77 to 83% of the loci. And there's only non-additive expression at 22 to 23% of the loci in the short liguled type and 16% in the long liguled type. So for the most part, there are not significant expression shifts or departures from non-additivity. And then finally, for those that did show any sort of departure, we investigated their gene ontologies. And so there were 39 loci that were overexpressed in the long liguled relative to the short liguled form, and only 2 in the reverse. And of these 39, 2 of them were actually associated with growth having to do with pectin and cell wall modification. And both of these would enable growth and would allow for the production of the larger inflorescence. And I will point out here that in these diagrams, we've drawn them to an equivalent scale, but they're on a real scale. The inflorescence for the long liguled is larger than the short liguled. So this provides us with some hypotheses about loci that could be investigated further. So to summarize all of this gene expression information, most of the genes have unbiased gene expression with a slight bias toward pretensis in both the long and short liguled forms. But most of the genes, also about the same percent, 80%, show the same expression pattern between the two forms. And most genes share expression patterns with their parents. 22% or 16% show non-additive expression, so where the expression patterns are like one parent or the other, or where they're transgressive. And finally, pectin and another cell wall component are more highly expressed in the long liguled form. So in terms of gene expression, are the tetraploid species of Trigopogon perfectly additive of their parents? The answer is clearly no. There is a lot of novelty at the level of gene expression, as well as gene content, as we saw with the homolog loss. And the final example here that I'd like to present has to do with the karyotypes. So, and this is work that Mike Chester did when he was a postdoc in our lab several years ago. So we would expect that in an allotetraploid, the diploids might cross and perhaps form a diploid hybrid as an intermediate step, but that ultimately when the allotetraploid is formed, we would expect this additive karyotype where the chromosomes are half from one parent and half from the other. So, of course, even Ownby's work showed a few departures from additivity where there were very rare cases of aneuploidy. And so when Mike began to examine these tetraploids with chromosome painting techniques, he also found a few examples of numerical aneuploids. So in this first panel, we see that there are 23 chromosomes instead of the expected 24, where there's been a loss of one of the E chromosomes. And in the second panel, 25 chromosomes instead of 24, with an extra chromosome, trisomy here for chromosome C of the pretensis subgenome. And these numerical aneuploids were quite rare, but we did detect them on occasion. What was much more common was something called compensated aneuploidy. And this is something that had been proposed in the literature and discussed in the context of crops, but something that was rarely, if ever, actually identified in nature. And this is a fascinating situation where in this individual at the top, it has the expected 24 chromosomes, but instead of there being half of the chromosomes from one subgenome and the other half from the other, we see, for example, trisomy of chromosome E from the dubious subgenome and monosomy from the pretensis subgenome. So there are 4 chromosome E's, but 3 of them are from one parent and 1 is from the other. In this next individual, we see monosomy of the dubious B, trisomy of the pretensis B. And here on the bottom, there is tetrasomy of the dubious E and nullisomy of the pretensis E. So there's a lot of variation in terms of gene or in terms of chromosome content, which I think clearly could be translated to shifts in gene content. So for some of those, so for an individual such as this one on the bottom, lacking both of the chromosomes from of E from pretensis, you could imagine that any of those genes would appear as homolog losses in that individual. And so these shifts in chromosome content due to compensated aneuploidy could in fact explain some of our instances of homolog loss. So this just summarizes the fractions of karyotypes that we observed in both Macellus and Mirus. This compensated aneuploidy was very common in Macellus, not quite as common in Mirus, where the majority of the individuals actually had additive karyotypes. But we see this compensated aneuploidy in both of them, and numerical aneuploidy is quite rare in both of these two tetraploids. So with regard to karyotypes, are the tetraploid species additive of their parents? Again, no, and there's a lot of novelty in terms of karyotypes within populations and among populations of these tetraploid species. Now we're in the process of assembling a dubious genome. This is some work that was done previously by Xiaoshan Liu when she was a graduate student with us, and we're now in the process of getting an even better assembly thanks to a collaboration with Dovetail Genomics. And so hopefully in the near future, I'll have some new genomic information that can provide some scaffolding for the rest of the work that I've been describing. In addition, Shan has also been developing CRISPR in Trigopogon. This was also part of his dissertation work, and he's continuing this as a postdoc now. And this is some work that was done in collaboration with Bing Yang, who was formerly at Iowa State University and is now at the University of Missouri and the Danforth Center in St. Louis. In addition, Shan has now perfected plant regeneration in Trigopogon. So in addition to being able to do the gene editing, we now can regenerate the edited plants. Here's both the diploid portfolius and one of the tetraploids, Mirus. So all of these things that I've described are pretty amazing changes that have occurred in a short period of time. Remember that these tetraploids originated since the early 1900s when they were introduced into the Pacific Northwest of the US. And so since that time, a lot of changes have occurred. So what's the state for some of these over longer periods of time? Is this all just sort of noise, or will some of this variation persist? Well, in order to get a handle on that question, we investigated an older tetraploid from Spain, and this is Trigopogon castellanos. We're not sure how old the species is, but we know that it's obviously older than the last 100 years. And in this tetraploid, we've conducted many of the same sorts of analyses that I just described. So I'm just going to show a snippet of information about these. So there are also multiple origins of Castellanos, one sort of in north central Spain and two separate origins in the Pyrenees. And for two origins, the one from north central Spain and one of the ones from the Pyrenees, they're perfectly additive in terms of their karyotypes of the parental subgenomes. But the third one shows some variations. So in this one, there are 4 chromosomes of the chrysopholia subgenome, no chromosomes of the Lamodii subgenome. So the same sort of compensated aneuploidy that we saw in Trigopogon mirus and Macellus has actually taken place here and has become fixed across this species. And there are also some translocations and things that are similar to things that we observed in the North American polyploids. So we see the same sorts of karyotypic changes, compensated aneuploidy and translocations. And we also see homolog losses, and these are more extensive than in the recent polyploids. And this is in a paper that actually came out yesterday in Frontiers. So another paper by Lucas Boatwright as the lead author. So a number of similarities to what we see in these younger species. So I think all of this indicates that we see a lot of novelty in polyploids. The polyploids are mosaics of additive, parental, and novel traits. There's variation among individuals, and this variation can generate arrays of novel combinations when these different individuals cross with each other. And we see this variation at many, many levels, genetic and chromosomal, but also cellular, physiological, and ecological levels. And I haven't shared any data on those latter components with you. But the polyploids, therefore, polyploid species are not composed of identical individuals. So this brings us to the point of what the evolutionary consequences of novelty might be. So if we think about variations such as what we observe in Trigopogon, it begins at the population level. And over time, this population level variation is manifested at deeper levels, somewhat deeper in phylogeny. And so we can start looking at older polyploidization events that have left signatures in the genomes of many plant species. And when we do that, we find that in fact there was an ancient whole genome duplication in the common ancestor of all living flowering plants. And this is dated to 192 million years. That's in line with some of the dates proposed for the common ancestor of all living angiosperms as well. So when the question arises as to what proportion of angiosperms might be polyploid, is it 30%, 50%, 70%? Actually, it's all of them. All angiosperms are polyploid on some level. And then there are subsequent duplications on top of these that mean that many species have undergone multiple rounds of polyploidy and that their genomes are the products of these repeated rounds of polyploidization. And this can be seen in this diagram taken from Cogipedia actually a few years ago. And what you can see here is everywhere where there's a star, there's at least one whole genome duplication. And so we see them in very key points in plant evolution. And I'll just focus here on a few of them. This one blue star and the common ancestor of flowering plants, that's the one I just mentioned. Two whole genome duplications in the common ancestor of this large eudicot clade, which contains about 75% of all angiosperm species. There are also duplications in the common ancestor of the monocots, and in particular in the common ancestor of the grasses. And in fact, when we start looking across at the locations of these whole genome duplications, we see that they tend to mark a number of large and well-recognized clades. And one of the examples here is the mints. The mints are the sixth largest family of angiosperms with over 7,000 species. And they've been the focus of the Mint Evolutionary Genomics Consortium. We've just completed a 5-year project, which of course raised more questions perhaps than it answered. And it was through this work that I got to know your own Benji Lichtman. And so for the next couple of slides, some of the work that I'll be describing was done in collaboration with him. So the Mint Evolutionary Genomics Consortium, through some transcriptome analyses, identified some key possible whole genome duplications that our lab then followed up on, where we're ultimately interested in the relationship among whole genome duplications, chemical novelty, and species diversification. Now, we found that the whole mint family is marked by at least one, possibly two, whole genome duplication events, and that across the family, everywhere there's one of these circles, there's evidence for one or more whole genome duplications. And the reason that there is this variation is that there are a number of different methods for doing these inferences. And so with the more conservative methods suggest 15 whole genome duplications, some of the other ones up to as many as 30. So this shows that there's been an incredible amount of whole genome duplication just within this clade. Now, Benji, along with our former postdoc Grant Godden and Robin Buell, who was the lead PI on the whole project, published this really nice paper last year on gene and genome duplications in the evolution of chemodiversity. And they suggested a number of areas where gene and genome duplications could have important roles relative to chemistry and also to species diversification. And we're currently in the process of following up on a few of these possibilities, which I will just mention here in the next slide. So one of our students, Alex Aber, has been working on constructing the phylogenetic tree for all species of mints. So he now has all species placed in this phylogenetic context, over 7,000 species. And eventually the next step will be to try to plot the whole genome duplications on this tree and equate that with chemical diversification and with species diversification, because it's our hypothesis that these whole genome duplications may have provided the fuel for new chemical pathways or chemical variants, and that chemical diversity may have driven species diversification within this clade. The other example is some work being done by Mallory St. Clair. Mallory was an undergraduate who just graduated with her bachelor's degree and who is starting a master's program with us this summer. She's focusing her work on lavender, again, studying selective sweeps and pan-genome structure, and eventually looking for correlations with chemical diversity. So whole genome duplications really come into play at deep phylogenetic scales, shallow phylogenetic scales, and with regard to species diversification and also trait evolution. So if we return to this view of whole genome duplications across the angiosperms, we can ask, are whole genome duplications associated with major diversification events? Because when we just look at that tree, that might be our impression. And it certainly was a hypothesis that occurred to us. So wanting to take a more critical look at this, we enlisted the skills of some comparative phylogeneticists who are much better at statistical analyses than we are. And we asked these questions. Is there perfect correspondence between ancient polyploidization and increased net diversification? Or is there a delay or lag in diversification following polyploidization? And the reason for this second question is that, in fact, it had been suggested, and in fact, our own phylogenies had shown that sometimes there's a whole genome duplication event, but it takes actually a few nodes into the tree before there actually seems to be diversification. So at the time that we conducted the study, which was in about 2014, we only were able to plot 9 whole genome duplications on the phylogeny for angiosperms, and then compare those locations with shifts in net diversification, where increases were shown in blue, decreases are shown in red. And when we do the analysis to look for correspondence between net increases in diversification and whole genome duplication, the only place where that was found was actually in the common ancestor of the Asteraceae or the composite. There's perfect correspondence there. In other cases, that there was not such a tight effect. So diversification shifts rarely are perfectly associated with whole genome duplications, but the shifts typically follow after a lag. So our conclusion here was that the whole genome duplications may promote, but are not sufficient to cause increased diversification. Now, of course, in that analysis, we were limited by the number of whole genome duplication events and also in our ability to place them with any confidence. So more recently, we've conducted another analysis. This was led by a former student of ours, Jacob Landis, and was published in 2018. Here we, all of the whole genome duplications are shown in green, increases in rates are shown in dark blue, and decreases in light blue. And for this analysis, we used a large angiosperm tree from Amy Zahn et al.'s paper. We plotted 106 whole genome duplications identified by Mike Barker and Zheng Li as part of the 1KP project. And we identified a number of shifts in diversification rate using multiple methods. And we found 41 of the 106 whole genome duplications were associated with upshifts rather than just the one that we found in the previous study. But, you know, that's not a significant value. But we did find that 66 of these whole genome duplications were associated with increased species richness in sister clade comparisons. So 2/3 or so actually show increased diversification, increased species richness relative to their sister groups. So are whole genome duplications associated with major diversification events? Well, sometimes they are. So 40% are statistically associated with upshifts, 67% have the higher species richness in sister clade comparisons. But so not always. But maybe what's more important is what happens when these whole genome duplication events occur. And these are changes, things that might occur at the genetic level that can then lead to phenotypic change. So we know that shortly after the common ancestor of angiosperms, where we see the whole genome duplication event, we get the origin of closed carpels, we get the derivation of vessel elements. Many of the key angiosperm features arise shortly after that whole genome duplication event. Likewise, in monocots, we see a number of phenotypic traits that arise. In the common ancestor of the eudicots, we see a shift to the pentamerous flower. We see the origins of new chemistry. And so it may be that these whole genome duplications, in some cases, trigger key innovations. So we can think about this whole process as a series that sort of combines our observations from Trigopogon and follows them hypothetically back in time to what may have happened to generate current patterns of diversification. So if we see polyploid formation happening across in multiple origins with these very rapid sorts of things that happen, as we see in Trigopogon, we can very quickly see polyploid diversity. Then following that diversification, if there's environmental filtering or drift, we may be left with a single genotype. If that genotype then encounters the right conditions, there could be a rapid radiation, which could be something comparable to what we saw for the Asteraceae. So we're using this as a model to think about how polyploidy can connect us from the microevolutionary to macroevolutionary scales. And ultimately, we think that, again, polyploidy allows us to think about cells to ecosystems, and it has impacts at biodiversity, biocomplexity, agricultural, biotechnological, and medical levels. And this is a paper we published last year with Don Fox as the lead author in Trends in Cell Biology, and it explores some of these themes in a bit more detail. So to summarize, Trigopogon has novelty that arises very quickly within polyploids at numerous levels, at the gene level, the expression level, karyotypic level, and morphological level. And polyploidy can also trigger diversification, with, again, 40% of the angiosperm whole genome duplications associated with upshifts in diversification, and 2/3 associated with increased richness in sister clades. And whole genome duplications may also lead to key innovations. Now, these impacts of polyploidization may be felt at the subcellular to the ecosystem scales, and they can have implications across many domains, from biodiversity to agriculture and to medicine. And I'd just like to acknowledge lots of folks who contributed to all of the work that I described today. And with that, I will stop sharing, and thank you very much for your attention. I'd be happy to take some questions.
Thank you. For questions from people at York, you could either just wave and I'll type them in the chat. So I'll take Kenchon's first. Oh, you're muted, Kenchon.
I was clapping my hands rather than putting my hand up, but I do actually have some questions.
Okay, go for it.
Okay.
It was a really fascinating talk and really exciting. I always find plant polyploidy to be really exciting, given that I work on butterflies and don't do that sort of thing. One question I had was about these independently derived tragopogon allopolyploids, and to what extent are they reproductively compatible amongst independent lineages, or does that not really matter because they're all kind of, they may have differing amounts of reproductive compatibility?
Oh, that's a great question. Thanks. And actually, I will point out that Mike Barker has at least proposed that there is a lot of ancient polyploidy in insects as well. So I think that there are hypotheses there to be tested. So some of your butterflies may be polyploid as well on a genomic scale. Okay, so back to your question. So yeah, that's a really excellent question. We set up a greenhouse experiment to test that very thing. But it's sometimes difficult to get things to flower at the same time. And I guess I'll just say we did not get enough crosses out of that experiment to be able to answer that question satisfactorily. But it's an excellent question and something that we've long been interested in. So these, we used to live in Eastern Washington, and these plants were literally in our backyard. And so it was very easy to grow them. The greenhouses handled them very well. The move to Florida was a bit more challenging. It was hot in the summer in Eastern Washington, but not humid, and the humidity really takes its toll. So developing the growth conditions here has been difficult, and we haven't been able to do as many of those sorts of experiments as we'd like to do, but still definitely on the list. So if we hopefully we'll get that answered one of these days.
And the other question I had was, how are these polyploids generated, particularly with respect to the compensated aneuploids that you were talking about?
So we think that we don't see very many diploid hybrids in nature. So we think that they actually form directly through unreduced gamete formation, so which can be caused by or stimulated by shifts in temperature and other environmental shifts. One of the things that's very interesting about where these plants occur as well is that it can be spring in March and it can be winter in May. So they can start bolting, they can produce start producing flowers by April or May, and then there might be snow, and then that may be a trigger for unreduced gamete production. So that could happen actually, simultaneously in the different species that are occurring together. So we think that diploid gametes of one may actually fertilize diploid gametes of the other, or they may go through a tetraploid or through a triploid intermediate, but we don't think that they form diploids, which then form tetraploids, because we almost never find diploids.
Yes, so I'm just wondering, if you had the unreduced gametes, so how are you getting the compensated aneuploids, where at one you get 4 copies of one and 0 copies of the other? Yeah, so we think that what happens there is that there are just some occasional pairing errors in meiosis, so that this is something that happens much later post formation, that maybe there's multivalent formation and you get like 3 going to 1 instead of splitting 2 and 2. Because as that compensated aneuploidy almost always is among the smaller chromosomes. I showed a couple of examples where they were actually on the B, but the vast majority of them are on the smaller chromosomes that are very similar in size and structure between the parental types. And they probably have the same gene content and can just substitute for each other. I have some more questions, but I'll let other people go first.
Okay, thanks for your questions.
I think Catherine had her hand up first.
Hi, thanks, Pamela. That was fantastic. So I was interested in when you're looking at the loss of homologs, either actual loss in terms of absence or expression sort of reduction, and just wondered, when it was non-random, have you looked at sort of what the function is of genes in those regions, and also whether those regions were then conserved in the Spanish older populations?
Great questions. Yeah, thank you. So we've tried to get a handle on what those genes might be, because you might have noticed in the figure we had a cluster diagram down the left side, basically. So we were interested in whether they were genes that were potentially interacting with each other. But the technique that we used for that analysis was something called a sequenome approach, so that we basically used a combination of amplification and sequencing to identify the SNPs. And they're very short pieces of DNA. So when we've tried to blast those through gene ontology, it's, we might get a huge category, but that's the best that we can do. So I think actually, despite how extraordinarily difficult that study was, we need to repeat it now with RNA-seq, so that, and even better with something like an iso-seq analysis, where we have very long transcripts where we can actually really figure out what those genes are, because it would be fascinating to find out. We know from other studies that there are that genes and polyploids interact in such ways that you can't, to basically retain stoichiometry within pathways. So certain genes are sort of resistant to being retained in duplicate, as long as the other components of their pathways are also not retained in duplicate. So it would be really interesting to find out what happens in those cases, and also to compare that with the Castellanos example. So yeah, excellent questions. I think it's another grant proposal, I think, that we need to work on. Excuse me. Thanks.
I think we have a question from Peter.
Hello, thank you. That was a super talk, and lots of really interesting questions. I just wanted to focus on one example that you spent some time on, and that was the Tragopogon species, which had long or short ligules depending on the direction of the cross. Mm-hmm. In this case, the maternal parent was the phenotype that was inherited. I wondered whether that was the normal rule, that it was the maternal phenotype that got into the hybrid. But more importantly, I was interested in what the mechanism might be for this. I mean, it's some kind of epigenetics, at least initially. Is it a difference in the organelle genotype, or is it a difference in inherited gene expression of the nuclear genome? Do you know what's going on?
No, we don't know what's going on. And that's why, those all of those expression examples, experiments that I described were really designed to just try to give us a bit of a handle on what might be happening. Obviously, it's not the organellar genomes themselves that are controlling that phenotype, but is there something about an interaction between the organelle and the nuclear genome that, through some of the physiological things that are taking place, that we might see some differences. So we kind of expected there to be differences showing higher gene expression in the one corresponding to the maternal genotype, and then the reverse in the other, and we didn't see any of that. So it's not something that is something you can examine through the expression level. So epigenetics sounds like a good target for that, but we really don't understand that at all. And the thing that's interesting is that Ownby reported this in his 1950 paper, and when we make them synthetically, we get the same phenotype. So it's there's something about the way that those crosses go that that really does generate that morphology, but what it is still a mystery. So yeah,
I don't know that we'll get to that one in our lifetimes. Maybe, you know, one of our students will have to pick that one up. But yeah, that's a really good question. Thank you.
Okay, thank you.
Benji.
Yeah, great talk, Pamela. It was really, really interesting.
Thank you.
You kind of mentioned at the end of the answer there about these synthetic polyploids, and they're particularly interesting. And so I was just wondering, when you're growing these, do you see any unusual properties of these plants within the first generation? And then if you're kind of crossing them down to further generations, do you see kind of fitness changes before they might lose some of these homologs?
So we multiple parts. Yeah, good questions. Multiple answers to that. So when we first treat them with treat either the seedlings or we use multiple methods to try to generate them. And early on, there are sometimes some, you know, very odd looking morphs that didn't make it very far. So where they just, you know, must have gotten totally messed up by having the colchicine treatment. But for those that actually did, you know, make it, and actually one other interesting thing is that sometimes we detected mosaics where the, you know, even certain roots might be diploid and other roots might be tetraploid. And that sometimes, you know, like the root shoot, there was variation between those. So there were a number of things early in the first generation that were sort of odd. But once they were established as lines, you know, they seemed to, we didn't notice general fitness differences, but what we did notice was that over time, we did some karyotypic work. We found that there were fewer abnormalities over even just 5 generations at the chromosomal level than, you know, as we went along. So that some of the things so there may have been some actual meiotic problems that were called out from one generation to the next, but we haven't done any analyses that actually would correspond to the homologs themselves. Well, we set up a big experiment, and there were too many things to be done in that. So there were some holes that ended up in the matrix, and the karyotypes really were part of that. But in this other experiment where we did look at them after like essentially each generation, we found a reduction in the chromosomal variation.
Great, thank you very much. I guess just curious about the method. So is this something that can work in other plants, or is it kind of it works particularly well for trying to go for your species?
It works. You know, people studying polyploidy in different systems are also generally able to make tetraploids synthetically. You know, one of the things that is emerging from people working on these different polyploid model systems is that some of them undergo homolog loss, some of them undergo karyotypic changes, some of them undergo gene expression shifts, et cetera. And not all of them do all of these things. And so there are a number of other of these polyploid models where we just don't see the karyotypic diversity at all. It's just very stable karyotypes across everything. So there's not really anything unique to Tragopogon in terms of methods, but it's a combination of applying the methods and there actually being the karyotypic or homolog type diversity, just because there seem to be different ways to respond to polyploidy, that there may be some underlying rules, but there's also a lot of diversity.
Great, thank you very much.
Yeah.
I have a question, actually.
Okay.
And early on in the beginning, you showed different populations of Tragopogon that didn't seem to be interbreeding. Do you think they're isolated because of the homolog loss?
So yeah, so we're not sure if they, how much of it is just gene flow opportunities
and migration of the fruits between populations, and how much of it might actually be either chromosomal or genetic incompatibilities. So I lied a little bit by omission. That top panel of Trichopogon mirus, the population at the very right side, which I think was either orange or blue, and there was one individual in it that was the other color. I don't remember which way it went, but anyway, there was one individual that was that was a different color, and that other individual had actually come from a nearby population, so it had the genotype of a neighboring population, which showed that there was at least the capacity for these fruits to travel and become established in other sites. But that was a, I would say it's a half a mile away. So maybe, a kilometer or something like that. And these are the inflorescences, well, when they fruit, are like large dandelions. And so each fruit has a very large parachute. They should be able to travel long distances. So we see that there is some possibility for that within one of these communities. But generally the formation occurs within disturbed areas within or right adjacent to a small rural community, and the next one might be 20 miles away. Okay. And so they don't really grow along agricultural fields or outside of these disturbed sites that are right around human settlements. So I think it's hard to get from one place to another. But then once they do, we're not really sure how well they can cross. And I think gets back to Kanchan's question earlier about crossability. So yeah, something we still need to find out.
Okay, so they could just look isolated because they're far away from each other.
Yeah, and they just haven't had opportunities to recombine at all. The Trichopogon miscellus has more populations and some of them are closer to each other. And that bottom panel had a bit more diversity within some of the populations, and some of the individuals also were more polymorphic than others. So I think they can cross if given the opportunity, but we really need to test that better.
Okay. I know I can see Kanchan has his hand up, but I think I might round us up now and thank Pamela Soltis for a wonderful talk, and to remind anyone that's on YouTube to keep an eye on us and our Twitter feed, because there'll be more. And I think we'll